Hi everyone, welcome back to my channel. Today I'm going to take you through the mass transport in animals sections for AQA A Level Biology. Also, I'll be going through some exam style questions and their mark schemes, and also, as always, I'll be putting in the comment section timestamps so you can skip to sections that you want to study from if you do not wish to watch the entire video. Right, so let's get started. So first we're going to talk about hemoglobin. Now, what is hemoglobin? Hemoglobin is this molecule here, and it is a globular protein consisting of 4 polypeptide chains. So I'll just get my pen and circle 4. As you're probably thinking, it has 4 polypeptide chains, so we can say that hemoglobin has a quaternary structure. Each chain, so we have 2 alpha chains and 2 beta chains, so 4 chains in total, and each one of these chains has what we call a heme group, and each heme group contains an Fe2 plus ion or a ferrous ion, shown by the red dots here. So each chain has one Fe2 plus ion or an iron ion. This property that it has the Fe2 plus bound to it means that oxygen can also bind to the hemoglobin. So we could say that hemoglobin has 4 oxygen binding sites. So the function of hemoglobin is to carry oxygen through the blood to respiring tissues for aerobic respiration to produce ATP. So now we're going to talk about the loading and unloading of oxygen to tissues. And we explain this via the oxyhemoglobin dissociation curve, which is what we have here. So first I'm going to talk to you about a concept called affinity. Affinity is basically another word for attraction. So we have this nice curve here, a sigmoidal shaped curve or an S-shaped curve. And as you can see, we have oxygen partial pressure on the x-axis and percent saturation of oxygen on the y-axis. Now at the beginning of the graph, at the low points here, we have a low oxygen partial pressure. So that basically means a low oxygen concentration. This means that there is less oxygen able to bind to hemoglobin, so it's less likely to bind. So we could say at this point, hemoglobin has a low affinity for oxygen, meaning that oxygen is less likely to load. And up here in this area, we have a higher partial pressure of oxygen. So this means that there is a high affinity as there is more oxygen able to load onto the hemoglobin, and this means that the saturation also increases. So now I'm going to explain about why the curve is S-shaped. So at the start here, it's kind of a slow start, a slow gradient or a low gradient. This is because there is a low partial pressure, so there's a low affinity. But as you can see, the gradient rapidly increases and the curve becomes more steep. This is because upon the binding of the first oxygen molecule, the tertiary structure or the conformation of hemoglobin changes. So the conformational change or the tertiary structure change makes the binding of the second oxygen and the third oxygen much easier, so the percentage saturation rapidly increases. Now, as you can see, the curve here starts to plateau or level off. This is because there's only one binding site left available, so there is a decrease in binding site availability, so it is harder for the oxygen to bind. And this leads to oxygen saturation when all 4 oxygen molecules have bound to the hemoglobin molecule. So the next thing you need to know about hemoglobin is what we call the Bohr Effect. So upon exercise, no matter what type of exercise, the rate of respiration rapidly increases because you are using more oxygen. This means that more carbon dioxide is produced, as carbon dioxide is a product of respiration, aerobic respiration. Now, when CO2 in the bloodstream dissolves in the blood, this forms carbonic acid, and as this is an acidic compound, carbonic acid, this lowers the blood pH, making the blood more acidic. This change in the pH, the lower pH, changes the tertiary structure or the conformation of the hemoglobin, so it has a lower affinity for oxygen. This means that oxygen is more likely to unload from the hemoglobin, so more oxygen dissociates at the respiring tissues to compensate for the higher rate of respiration that your body is carrying out through exercise, obviously providing an advantage as more ATP will be produced. So this is how we represent the Bohr effect on the dissociation curve. Now the dotted lines here are very faint, but hopefully you can see them. So when the hemoglobin has a lower affinity, the dissociation curve shifts to the right. I'm kind of roughly tracing it here because it's a very faint line. That's a very bad line, and apologise for that. So when the affinity decreases, the curve shifts to the right. So this is what the curve would look like for the Bohr effect. Now, if the hemoglobin has a higher affinity, then the curve shifts to the left, as there is a higher saturation at lower partial pressures. But when there's a lower affinity, there is a lower saturation at higher partial pressures. So different organisms have different hemoglobins, types of hemoglobin, or different hemoglobin structures. So here's an example. Mountain dwellers, for example mountain goats, as shown in the image here, have a hemoglobin with a higher affinity for oxygen, so oxygen is more likely to associate or load onto the hemoglobin. This means that the hemoglobin is loaded with oxygen at low partial pressure. This is important for mountain dwellers as there is often less oxygen available at higher altitudes. So having the hemoglobin with higher affinity means that the few oxygen that is there binds to the hemoglobin, as the hemoglobin has a higher affinity. So the next example is organisms such as fetuses, worms, especially lugworms, have some myoglobin, which is a different kind of protein, which has a higher affinity, so oxygen is more likely to bind. So here I've just got an image of a lugworm. Right, now I'm going to get onto the circulatory system of mammals. So here is a nice image that I stole from the internet of the basic circulatory system of mammals. Now the key facts you need to remember here are veins, which are a type of blood vessel, go towards the heart, and arteries travel away from the heart. We can therefore conclude that veins contain deoxygenated blood and arteries contain oxygenated blood. Now, the way that I like to remember this is one of my teachers taught me this. Vein kind of sounds like vein in, so it's a vein in towards the heart. Kind of sounds silly, but it's a good way to remember it. So here we have various blood vessels. Now you need to know the names of the vessels that go in and out of various organs. So here we have the blood vessels surrounding the liver. So the blood vessel going towards the liver, entering the liver, is called a hepatic artery. Remember this term here, hepatic. And the vessels leaving the liver are called the hepatic vein. And for kidneys, it's basically the same principle, but we use the word renal instead. Here we have the main blood vessels supplying the heart. So the blood vessel that supplies the heart with deoxygenated blood is called a vena cava, which is a vein. And then the vessel that leaves the heart and goes towards the lungs is called the pulmonary artery. noting that this is still deoxygenated as it hasn't reached the lungs yet. Then the blood reaches the lungs, which is then oxygenated through the process of breathing and diffusion into the capillaries, and then the blood enters the pulmonary vein, which then enters the heart, and then through the atria and the ventricles and into the aorta, which is the main artery that supplies the whole body. So now we're going to look a bit more about the human heart, or the structure of the human heart. So here we have the vena cava, which is from the upper body, and the other vena cava, which is from the lower body. So deoxygenated blood from various organs enters the right atrium from the vena cava. Now, some people sometimes get confused between the left side of the heart and the right side of the heart. Now, you've got to imagine the picture of the heart like it's on your own body. So if you're looking at an image of the heart, the side that's on the left to you is the right side of the heart, but the side that is on the right to you is actually the left side of the heart. So deoxygenated blood enters the right atrium first, which is one of the 4 chambers of the heart. Then the tricuspid valve opens and the deoxygenated blood enters the right ventricle. Now, the function of the valves here is to prevent backflow from the ventricle to the atrium. We will delve into more detail in that later. Then, the deoxygenated blood enters the pulmonary artery. The blood is then carried to the lungs where it is oxygenated. And the oxygenated blood then enters the left atrium via pulmonary veins, and then enters the left ventricle via bicuspid valves. It's important that you don't get confused between bicuspid and tricuspid. There's not really a way to remember it, so you just need to manage to memorize this. The left ventricle blood then enters the aorta, which is then pumped around the whole body. Now, some key adaptations here is that the walls of the left ventricle are much thicker than the right ventricle. This is because the oxygenated blood from the left ventricle has to be pumped around the whole body, so a higher muscle power is required because a higher pressure is required as the blood is being pumped around the whole body instead of just to the lungs. So the atria and the ventricles are different in terms of their properties as well. So the atria are much thinner than the ventricles. This is because the job of the atria is to only pump the blood to the ventricles. However, the job of the ventricles is to pump, well, in the right ventricle case, to the lungs, and the left ventricle case to the whole body. And also we have here, which is this bottom vessel here, which is called the coronary artery. The job of the coronary artery is to carry oxygen to the heart muscle. This ensures that the muscle can contract properly to pump blood. So now you need to know in a bit more detail about the cardiac cycle, so the volume and pressure changes associated with this. So, the first step in the cardiac cycle is what we call cardiac diastole. In cardiac diastole, the atria and the ventricles are relaxed and the blood enters the atria, so you can see the blood is entering here. This is the right atria and this is the left atria here. So the blood enters the atria. This increases the volume of the atria and therefore increases the pressure. So as the blood enters, the pressure rises. So the AV valves, this means the atrium ventricular valves, so the bicuspid and the tricuspid valves, open because the pressure is high. This means that the blood flows into the ventricles down a pressure gradient. So the ventricles volume then increases. So the next step is atrial systole and ventricle diastole. So in this step, the atria contract when they are about 50% empty. This ensures that all of the blood has entered the ventricles. When blood enters the ventricles, this is called ventricular diastole, as the ventricles have to relax to allow blood to fill them. Now, as the blood enters the ventricles, the ventricular pressure slightly increases. As this pressure exceeds the pressure in the atria, the atrioventricular valves, so the bicuspid and tricuspid valves, shut to prevent backflow. So the final step is ventricular systole. Ventricular systole is when the ventricles contract. They contract harder than the atria as they have thicker muscular walls. This further increases the ventricular pressure, so it's more than that of the pulmonary artery in the case of the right side or the aorta in the case of the left side. This means that the semilunar valves open. I didn't mention this earlier, but the semilunar valves are the valves between the ventricles and the pulmonary artery or the aorta. So these valves open so the blood flows into the pulmonary artery or the aorta. So an important adaptation of the circulatory system in mammals is this double circulatory system. This is an important adaptation as it allows a high pressure to be maintained. A single circulatory system wouldn't work in us because when the deoxygenated blood travels to the lungs, it enters capillaries. Now, the surface area of capillaries is huge, so the large surface area of the lung capillaries would decrease the pressure, as there is a large surface area, so a higher volume. This would mean a decrease in pressure, so less oxygenated blood would be delivered to the tissues, which would obviously be detrimental to respiration rates. So now you need to know about the adaptations of different types of blood vessels. So first we're going to talk about arteries. Arteries contain a thick muscular layer. This means that constriction and dilation can control blood volume so that the correct amount of oxygen is delivered to tissues. Arteries also have a thick elastic layer to maintain blood pressure and to allow stretch and recoil. The maintenance of blood pressure is important for the same reason that I mentioned previously. And stretch and recoil also maintains blood pressure and allows the maximum oxygen and nutrients are delivered to tissues. Also, the arteries have a very thick wall to prevent bursting due to high pressures within the arteries. So now we're going to talk about arterioles, which I haven't yet mentioned. So arterioles are these blood vessels that branch off from the arteries, so this kind of areas here. Now, the job of the arterioles is to restrict blood flow into the capillaries. So they're kind of the connection between arteries and capillaries. Now, arterioles have a thick muscle layer to restrict blood flow into the capillaries. This is important as it allows the control of the blood flow so that not too much blood enters the capillaries, so that would mean the capillaries would burst, as capillaries are very thin. Also, they have a thinner elastic layer and wall than arteries. So now we're going to talk about veins. Veins have a thin muscle layer, have a thin elastic layer and wall due to the lower pressure, as they are going towards the heart and not away, so they don't need to carry blood to tissues. And also, another adaptation is the thin layers that I have mentioned mean that they can be flattened easily, helping blood flow to the heart as there is a larger surface area. Also, they contain valves to prevent backflow. Now, the importance of valves is that, obviously, I've mentioned that there is a lower pressure. A lower pressure means that backflow is possible. This is where the valves come in, so they prevent the backflow, so therefore preventing the possible mixture of oxygenated and deoxygenated blood. So the last type of vessel we need to know about are capillaries. Capillaries have no muscle or elastic layer. They have a wall that is 1 cell thick, so they are very, very thin capillaries, or they are very small. And the clever thing about capillaries is that their small diameter and their small size means that they can perform these extensive networks, which is shown in the diagram here, called capillary beds, which have a narrow diameter. This means that the red blood cells travelling through the capillaries are often squashed against the wall. This means that there is a short diffusion distance of oxygen into the capillaries. The wall that is 1 cell thick also has the same adaptation as this, so there's a short diffusion path of oxygen into the capillaries. This means that oxygen can be readily unloaded at respiring tissues. So the last thing that you need to know for this topic is the formation and return of tissue fluid. This shouldn't be this off. So what is tissue fluid? So when blood enters capillaries from the arterioles, there is an increased volume inside the capillaries. This results in a high hydrostatic pressure, so a high pressure of liquids. This is at the arterial end. Now the arterial end is is kind of like the start of the capillaries. So when the arterioles branch off into capillaries. This high hydrostatic pressure means that molecules like glucose, amino acids, fatty acids, ions, water, and oxygen are forced out of the capillaries into the surrounding area. This is what we call the tissue fluid. So the tissue fluid is a mixture of all of these kinds of molecules here. Now the tissue fluid supplies tissues with these nutrients and oxygen. But larger molecules, such as proteins, remain in the capillaries and they are not forced out of the capillary as they are too large. The presence of the proteins remaining in the capillaries mean that the water potential is lowered. So that there is a lower concentration of water in the capillaries at the venule end. So this means that at the venule end there is a low hydrostatic pressure now, as there is less liquid and a lower water potential. This means that the water must re-enter by osmosis, as osmosis is the movement of water from a high to a low water potential. So water moves into the capillaries at the venule end because there is a lower water potential there. However, not all the tissue fluid enters here. The remaining tissue fluid is absorbed into the lymphatic system, where it is later then reabsorbed into the capillaries. Now this normally happens at the capillaries near the heart as well. Right, so that is it for the content, and now we're going to get on to some exam style questions. The highlighter. So let's look at this first question. What is the function of coronary arteries? Now this is a 2-mark question that I've cut off. So 2 marks for this question. So as I said earlier, the function of coronary arteries is to carry oxygen to the heart tissue. So carries oxygen to the heart muscle. A pretty simple question. So let's look at the mark scheme. So the mark point 1 is it carries oxygen, or you can write it carries glucose. So we'll get that mark. So it says accept oxygenated blood as this contains oxygen, so you can get the mark if you wrote that. But it says ignore references to removing waste products and ignore references to arteries pumping blood, as these aren't relevant to the function of the coronary artery. So if you put any of those, you don't get the mark for that marking point. Now the second marking point is to the heart muscle, tissue, cells, or myocytes. Now as the words muscle, tissue, cells, or myocytes are underlined, you must use one of these words to get the mark. So if you just put carries oxygen to the heart, you wouldn't get the second mark, as you need to refer to the heart muscle or tissue. So we would get both marks there. So let's look at this question. Figure 1 shows some of the large blood vessels in a mammal. So this is a simplified version of the figure that I showed you earlier. So this is quite a nice question. So which of the blood vessels A to H is the vena cava? So as we said earlier, the vena cava is the vessel that carries deoxygenated blood into the heart. So as you can see here, A is the blood vessel that carries blood to the heart, deoxygenated blood. So let's look at the next part. Which of the blood vessels is the renal artery? Now, as I said earlier, renal is to do with the kidneys, so renal means kidneys. And when we're talking about organs that are not the heart, arteries are the things that leave the organ, not enter the organs even. So H is the renal artery. So A, we got that right, and H, we got that right. Now it says here, accept A on its own even if outside box. Now if you are anything like I was at A level, I change my mind a lot, so my answer often doesn't end up inside the box. So you still accept it even if it's not in the box. Now it says reject if 2 or more letters given. So if you put A or D, for example, you don't get any marks, you just need if one of them is wrong. So let's look at the next question. Figure 2 shows how the blood pressure changes as blood travels from the aorta to the capillaries. The rise and fall in blood pressure in the aorta is greater than in the small arteries. Suggest why. Now, when I was talking about the structure of the heart earlier, I said the aorta is a vessel that supplies oxygenated blood to all of the body. So here is what I have put. The aortic pressure is much higher, as you can see by the graph here. So the pressure is much higher than the small arteries. This is because, as I've mentioned earlier, it has elastic tissue and stretches and recoils. So you need to kind of link in the properties of different vessels into this. Now, as the aorta is an artery, we can say the elastic tissue has stretch and recoil properties. So let's look at the mark scheme. So the aorta is close or directly linked to the heart, the ventricle, or the pressure is higher or is very high. So we'll get that mark as we put this option here, the pressure is higher. So the second marking point here is that the aorta has elastic tissue, which we wrote, and they accept elasticity, but they ignore reference to the muscle. And the final marking point is aorta has stretch and recoil, which we wrote. Now they reject contracts, relaxes, and pumps. This is not what the aorta does, that is what the ventricles do and the atria do. And as it says reject, if you wrote this in your answer, you don't get any marks at all for the entire question. So even if you got these 2 marking points correct, you wouldn't get any marks if you wrote this. So let's look at this next question. The figure below shows a capillary surrounded by tissue fluid. The values of the hydrostatic pressure are shown. So here at the arterial end, we have a hydrostatic pressure of 4.3 kilopascals. And at the ventral end here, we have a hydrostatic pressure of 1.6 kilopascals. So the question says, use the information in the figure above to explain how tissue fluid is formed. Now, as I said earlier, tissue fluid is formed when the hydrostatic pressure means molecules are forced out of the capillary. Here's what I've put. The outward pressure, so the pressure from the capillary to the fluid, is 4.2 kilopascals in total. Actually, I should have put 3.2, that is a typo, I'm sorry. So the outward pressure is actually 3.2 kilopascals, which was a typo that I have made, sorry. As the outward pressure is positive, this forces small molecules out of the capillary into the tissue fluid. So let's look at the mark scheme. The overall outward pressure is 3.2 kilopascal, so forces small molecules at the capillary. Easy 2 marks there. So let's get on to the next one. The hydrostatic pressure falls from the arterial end of the capillary to the venular end of the capillary. Explain why. So here I've already printed the mark scheme. So water exits the capillary at the arterial end. Now I put my answer relating to water as hydrostatic pressure relates to the volume and the pressure that the water brings. And I've said at the arterial end as this is where the water exits. Now I don't really need to write much detail as this is only a 1 mark question. So the mark scheme says loss of water or loss of fluid or friction against capillary lining. Now I haven't put the exact words of the mark scheme, however I've got the ideas right, so I would get that mark. And none of the marking points here are underlined, so we don't need to write the exact phrases used in the mark scheme. So let's look at this question. High blood pressure leads to an accumulation of tissue fluid. Explain how. Now, as this is an explain question, you don't just need to write what is happening, you need to write why it is happening. This is quite a nice question. It basically just wants you to write how tissue fluid is formed. Now, this is what I've put. So there's a high hydrostatic pressure due to the high blood pressure. This is a key term that the examiners like to use or like you to put when there is a question about tissue fluid. The high hydrostatic pressure due to the high blood pressure from the blood entering from the arterioles. This creates an increased outward pressure from the arterial end. Now the key term here is outward pressure, which I didn't mention when I was talking about tissue fluid earlier. So make sure you put outward pressure, and that is from the arterial end. This means that more tissue fluid is formed as the molecules and the water are moving out of the capillary. So here's the mark scheme. High blood pressure means a high hydrostatic pressure, so you would get that mark. This increases the outward pressure from the arterial end of the capillary, which we wrote, or you can write it reduces the inward pressure at the venular end of the capillary, which is the converse of this point here. So you can get the mark if you wrote that as well. So this means that more tissue fluid is formed or less tissue fluid is reabsorbed. So we get all 3 marks for this question. And it also says here, allow lymph system not able to drain tissues fast enough. So you can get the mark if you wrote stuff relating to the lymph system as well. Right, let's go on to the last major question of the video. The figure below shows recordings made from the heart of a dog. So this top recording here shows the pressure in the ventricle. The middle one shows the blood flow into the aorta, and the bottom one shows the thickness of the ventricle wall. So they are very different measures. So the first part of the question says, use information from the figure to explain how the pressure in the dog's ventricle is related to blood flow into the aorta. So in your answer, you need to interrelate these 2 things. You can't just talk about them separately. So this is what I've put. Ventricular pressure rises. As you can see from the graph here, it rises. Then blood flows into the aorta as the pressure causes the semilunar valve to open. Then the ventricular pressure falls, so blood flow decreases. So I have used information from the figure. As I've said, the pressure in the ventricle increases and the pressure then falls and the blood flow decreases as the blood flow decreases here when the pressure decreases in the ventricle. So let's look at the mark scheme. Ventricle pressure rises, then blood flow starts to flow into the aorta because pressure causes the aortic or the semilunar valve to open. So it accepts the time, so you can use values from the graph, but I think it's easier that you don't use that. And it says here, idea of sequence is essential. So you don't refer to these as separate things. You need to link them together in a sequence to get marks so that the examiner knows what you're talking about. And the second marking point is the ventricle pressure starts to fall, so blood flow falls. And again, the idea of sequence is essential here. So if we look at the next one, use the information from the figure to explain how the pressure in a dog's ventricles is related to the thickness of the ventricle wall. Here's what I've put. The thickness of the ventricle wall increases, as we can see here. There is a rapid increase in the thickness of the ventricle wall. Now you need to relate this to what happens during the cardiac cycle, which I mentioned earlier in the video. So the thickness of the ventricle wall increases because the ventricle wall contracts. So the contraction causes the increase in pressure. So you need to relate it to the pressure in the ventricle, as this is what the question asks you. So as you can see from the recordings here, when the thickness of the ventricle wall increases, the pressure in the ventricle increases. So you have to write that in your answer as well. So the thickness of the ventricle wall increases because the ventricle wall contracts, so the contraction causes the increase in pressure. So here's the mark scheme. The thickness of the wall increases because ventricle wall contracts. So we get that mark here. I need to provide the explanation in the terms of because the thickness of the ventricle is a direct result of the contraction. So here it says, must be idea that increase in thickness is linked to contraction, so you need to link them somehow. Also it says, except muscle ventricle and systole for muscle contraction. So the second marking point is contraction causes the increase in pressure. So the increase in pressure is a direct result of the contraction. And it says except thickening of the wall. So you can write that as well if you want. So that's 2 marks there. So this is the last question that we're going to talk about, which is a maths question, which is everyone's favourite. So use a figure to calculate the heart rate of the dog in beats per minute. Now here I've looked at this top graph, and as you can see it measures 2 seconds. And I have calculated that there are 4 beats. The distance of a beat from this recording, if I get my pen, is the distance from the trough, which is the lowest point in the pattern, to the next trough, so it's here. So we can count 1, 2, 3, 4 troughs here in 2 seconds. So there are 4 beats in 2 seconds. You can also count the number of beats by the number of peaks. So 1, 2, 3, 4 in 2 seconds. So 4 beats in 2 seconds. Now, as the question says beats per minute, we need to calculate how many beats there are in a minute. So 60 divided by 2 is 30. So there are 32 seconds in a minute. This means that we need to multiply 4 by 30 to get the number of beats per minute. So 4 times 30 is 120. So we can say there are 120 beats per minute. Let's look at the mark scheme. So the first marking point is between 120 or plus or minus 5. So this means that you can have your answer in the range of 115 or 125. So if you got this answer right, you actually get both marks. So we get both marks. And the plus or minus 5 here is because the length of the cycles varies slightly in each recording, and it can be subjective depending on your eyesight. So here it says below, the length of the cardiac cycle correct, but the final answer is wrong. So if you got the length of the cardiac cycle correct, which you might have calculated to be 0.5 to 0.52 seconds, if you want to be that specific, but you got the final answer wrong, you can still get a mark. Right, so that is all I want to say. Thanks for watching. Please comment below if you have any questions, and I'll see you in the next video.